Over to, I guess, straight over to you. Is Wolfgang going to say a few words? And probably not. So, okay. Ewan, over to you, and thank you all very much. Hope you have a great day.
Thanks, David.
Thank you. Thanks. So let me give my welcome, and I think the welcome on behalf of the organisers, so Peter, Wolfgang, and Richard. Your two communities here, it's very deliberate. Some of you should be, I hope, statisticians. Those are the people who I don't really know. So hello. And then some of you are going to be more biologist, of a biologist bent. Because of that, there are going to be no stupid questions in this meeting. It's official. It's impossible for anybody to ask a stupid question, because on each side of that there will be obvious starting points that half the audience will understand and the other half won't. Some of this, quite deliberately, if you haven't seen the way the Newton Institute works, there are blackboards everywhere, literally, in the Newton Institute. So as you go out, there is a blackboard wherever you see it. And this is for you to grab someone who you did not understand precisely what they meant when they talked about it in coffee. You earwigged a conversation and you said, I don't quite understand that. You can grab them, drag them to a blackboard and say, please explain what is going on here. And the goal is to create more collaborations and cross-fertilization between these two fields, which I think are very, very relevant to each other and also very interesting pieces of science from both perspectives. And the goal of us doing this workshop was deliberately to catalyze this kind of cross-fertilization. So I think that's my leading set, and I'm the first speaker, and I'm going to speak about some work on mammalian promoters, and this is mainly the work of my student Michael Hoffman, who is currently touring the States at the moment to look for postdoc positions effectively, and it's been great working with him. But this is going to be about the properties of natural selection and mammalian promoters. So let's see if I can do this. So, which one's the zapper? No. Click. Okay, this is too complicated for me. It looks like it should be a laser pointer. Does somebody know how that thing works? Do you want to play around with that whilst I draw? First thing. Okay, so there are three parts of this. So one of them I'm going to not dwell too much on, because in fact we have created a probabilistic model of transcription factor binding. It's very similar in many ways to many other approaches, but I am going to go through it so that you understand what on earth works. Oh, that looks good. That's more obvious. Okay. And then the second two parts of this are the more interesting parts. So the model that we have for transcription factor binding is called Sunflower, so that's the name of our model. And then we're going to use that, I think, in an interesting way. So understanding how mutations change this process, and understanding, therefore, how to think about promoter evolution. Now, quite possibly, I've already lost half the audience in that very first sentence. So this is a very quick crash course in DNA binding, and you'll get this a bit more from Martha Bullock as well later on. So DNA, we think of it as a linear sequence, and these have letters underneath this, A, T, G, and C. Those letters are actually chemical structures, but in all the analysis that we do, we just treat them as letters. And what's known in biology is that proteins bind regions of DNA, and that binding is mediated by an interaction between the protein and the DNA sequence. And though it's not obvious actually with this screen, I've drawn these in slightly different colors. So here are two proteins, the square and the circle, and they're binding at different affinities. So these are darker red than these ones here. And so this is a particular binding of these two proteins where 2 copies of the green and 4 copies of the red bind this DNA sequence. Now what's known is that this process, or it's pretty much known, is in fact a stochastic process, and one can certainly think about this in a very biophysical way. That is, that there are different energy states, or one can think about this, as I'll come on to say, in a probabilistic way. So here's a different binding configuration, precisely the same DNA sequence, And here, instead of this red thing, we have a green one here binding instead of this. So these could be two different states of the system with two different energies at the end of the day. And one can get into another, this would be another state. In this case, this green box, which should be slightly less green, is binding instead of this higher affinity red box. And because there's a slight overlap, these are mutually exclusive. Either this binds or this binds, but not both of them at the same time. Probably this would be at a higher energy state because this is a lower affinity binding in the schematic that I've done. Now you can imagine for a particular DNA sequence and with a particular set of factors, if you consider all these different possibilities, you have a lot of different possible states. So I can now think about the red one being 1 to the left, 2 to the left, there being no red or no green, different combinations of all these things. All of those possible scenarios exist in biology, and we want to model those. So the way we model that is a very standard sort of way. It's a hidden Markov model. And this model has states now where each of the, so this model we call sunflower because it looks a bit like a flower. Here's the central state, and here's these petals. And each state here represents the binding of a particular transcription factor. And so once one goes in at a particular site, one will go through a series of states and then return to this silent state. And there's a kind of catch-all unbound state that represents the gaps which are not bound in this system. And that's the fact that you can only be in one of these states implicitly handles the mutual exclusion of transcription factors. And for each of these transcription factors, one has some model of the affinity of binding. Now this is one of the classic proteins DNA motifs. This is called the Tata box because it goes T-A-T-A, and there's a kind of T at the end. So this is a binding affinity of a particular protein. Now, at the moment, we're just going to take this as given that we get a list of these things. We're going to take actually 70 cases. That number 70 is far smaller than the number that's in biology. The number that is in each of our cells is about 2,000 proteins that have this. There are about 70 proteins where we have a model of its affinity. And then we take a model of this affinity, and this becomes, in this case, a 14-state petal. So here it would be coming in from the silent state. We go in here, and we go through all 14 states. Kind of critically, once you've decided you're going into this state, you have to go through all of these 14 states, and then you return back to the silent state. So that's our kind of model. And for the aficionados, so that's, again, each one of these petals represents now one of those logos. And we're going to have about 70 of these petals going around this. Now, this model is not tremendously new. A number of different groups have done very similar things. So Aaron Segal's group have done this. Martin Bingorn's group has done this. And the group, I forget his, Bussamonkey, or no, guy from New York, and the program called AHAB has done this. So these models are all very similar. Sometimes they use a biophysical framework. We use the parameterization based around probabilities, and it's really quite equivalent. You can use either framework. And so with this framework, we can use very standard hidden Markov model technology to get numbers out. So we take the hidden Markov model and run forwards, backwards. Now I may have lost the other half of the audience. But what forwards, backwards method allows you to do is sum all the possible states. So all of those different configurations where you have red, 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 green, and then you go green, red, 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 and then you place them into all these different possibilities. So you can go over all those possibilities and get out marginal posterior probabilities on each base in the sequence. And we could have 70 times the length of the transcription factor marginal probabilities, but because when you enter on the 0th state, you're kind of forced to go all the way through, we're only really interested in the marginal probabilities of the transcription factor on the 0th state. So I'm going to show lots of plots now, and in general, we just show the posterior probability of the 0th state, the first state in the transcription factor. And so for each base of the DNA sequence, we end up with a vector of, in our case, 70 numbers plus one more number, which is the unbound state of the marginal posterior probabilities that that base is bound by this factor, or this factor, or this factor, or this factor, or this factor. Now, just to stress, you can map these back to biophysical models if you like, and there's some kind of recipe and debate about precisely how you do that. From this perspective, the only interesting thing is we can get out this vector of numbers. So just to show you this graphically, this is now for one transcription factor on one region of the human genome. This is, in fact, the Tata binding protein, and this is a region of the human genome which is centered around the start point of this gene here. And I'm plotting now the posterior, Michael's plotting the posterior probability of the entry into the zero state. And you can see here there's a really nice peak that goes all the way up here, which is nicely just in front of 0. This is what you expect because a gene like this has a Tata box just above 0. The key thing is that we can now do this where we run all 70 factors simultaneously. So they're all in competition with each other. They're all trying to sterically compete with each other for the same bits of DNA. And this now is the same plot now for that same promoter, but now with 70 factors. So in each case here, we have 1,000 positions going across here, and now we have 70 numbers per each 1,000 positions.
Question?
Sure?
Stupid question.
No stupid questions. It's officially impossible.
So this is posterior to what?
Sorry?
What is X?
Given X, the DNA sequence. So we are given the DNA sequence. So you have this model of transcription factor affinities, and you have a given DNA sequence.
So for a given DNA sequence, and your prior probabilities are the motif
Affinities, yeah.
that you are given.
That's correct.
Databases from accumulated biological knowledge.
Accumulated biological knowledge, in particular, there's a set of experiments to do this. There's one thing that I haven't mentioned here. We have to set this parameter here, which is the entry parameter into the transcription factors. And you could do this, arguably that parameter represents the concentration of each transcription factor. Okay? You could perhaps do something fancy with gene expression data for a particular cell type. We, at the moment, have set a single flat prior, and we've set that prior that gives us results that we like later on in the process. Not a brilliant way of setting that prior, but you know, it's a number that we have to pull out of the air at the moment, which is whatever this number is.
Have you actually looked at expression data to see how flat they are?
We haven't directly ourselves. Because later on, we'll come on to this, because later on we want to use this for a model of evolution, there's a little bit of a theoretical thing that we want to do it across all cell types.
Generalizability and so on.
Yeah.
But for a specific cell
We haven't looked at, people have, we haven't looked particularly.
So for clarification, I just wonder, so you said zero state basically means the TF binding start site?
Yes, the zero state, I mean, it all depends whether you like to number things from 0 or 1, and Michael likes to number things from 0. So it means the first state, but he's Python obsessed.
So implicitly, you're still somehow thinking of things binding independently position to position, because you say there's a certain probability of that particular transcription factor binding, and then given that that's the case, what's the probability that you
Yeah.
that particular base?
So this has independence of all the transcription factor binding to each other, except for the fact that they can sterically hinder each other. So in the configuration, if a particular site is particularly strong for a particular transcription factor, it forces out other weak sites nearby. It will downweight those posterior probabilities. And so even with, as I'll come on to show, even with this single sort of addition of steric hindrance, we get quite complex effects coming out of the process. Wolfgang?
What's the implication of using the same initial probability for all of the 72, for all of the 80?
All of these guys, yeah. It's as if we're saying there's a flat concentration of all the transcription factors. We haven't investigated that in detail at the moment, okay? So obviously one can play with that. Okay, so, oops, now I've gone too far. Right, so that was with one transcription factor, one sequence, sorry. That was just with one DNA sequence of this particular gene. We now do this on 17,000 sequences. We've said all human transcripts. What we mean there is all human transcripts where we're relatively certain that we know where the transcription start site actually happens, so we can get the registering correct. So there's 70,000 transcripts where we're reasonably confident that this is where the transcript starts. And this now is the sum of that probability for all the different factors. And you can see there's quite a variation factor to factor. Very nicely here, here's the tartar binding protein. The people at the front can see that there's a very nice kind of flat and a very sharp peak very close to 0. Notice there are these guys here that show quite a strong humpy kind of binding. Now, this is going to come through my talk a little bit. The humpiness will be the well-known annoying effect of CpG islands, basically. So again, for the non-biologists in the room, there's a kind of quirk of biology such that CpGs, C followed by G, are suppressed generally in the human genome. The only places where they're not is where there's consistent unmethylation of the DNA sequence, and that often happens on promoters of genes that are used a lot, so-called housekeeping genes. So housekeeping genes have a higher level of GC generally, so they have more Gs and more Cs, and specifically a higher level of C followed by G. And that's just the way it is. And here what we've done is we split all 17,000s into two classes. CpG islands, those containing these high GC, CG-rich regions, and CpG deserts, which is basically everything else. And you can see that the transcription factors vary in their response to those things. So again, the people at the front notice that TBP, this peak, is nearly always in CpG deserts. It's not in CpG islands. And again, that agrees with some pieces of known biology about how this works. Now, this 70-fold plot kind of makes you feel that the green line is always above the red line. Just to say that's not the case. If you focus in on certain transcription factors, you can see here that there are some transcription factors that definitely we predict like CpG deserts a lot more than CpG islands. I'm going to return to this point at the end, but quite interestingly, if you just take this data, now on the y-axis here, I kind of have its relative CpG islandness. So up here are the guys that really like to bind to only CpG islands, and down here are the ones that really don't like to bind here. And then here is just the total binding. And you can see here that our predicted there's a big correlation here between the sort of total binding amount and whether you like to bind CpG islands or not. And furthermore, there's a big correlation. This is again the total binding, and then this is replacing here on the y-axis just the GC content. So this is not even the CpG, predicted CpG content, just the GC content of the binding affinity, position weight matrix, the affinity of the transcription factor. So actually up here are your classic housekeeping, SP1 is up here, for example, which is a very general transcription factor. Very interestingly, down here are a whole bunch of factors which are more traditionally associated with developmental genes. So it's not the case that, as well as there being this correlation seems to be correlated with something interesting as well, just as like an extra irritation. So I'm going to come back to this point again at the end, but just to say that we already observe some sort of interesting features, these things. Now this is, it's kind of trivial. We can do this on 1,000 base pairs. We can also do this on the whole genome. One has to be a little bit sneaky about how one stores the numbers, because 3 times 10 to the 9 times 70 sets of numbers is quite a lot of numbers. So one just has to be a little bit clever about that. But here we have the binding site of the tartar binding protein across the genome on this model. And another thing which I won't mention, but if you want to grab her, Alison, who can now wave, there she is, who's another student in the group, she has extended this model to put cooperativity states in this. So this is homotypic cooperativity. Cooperativity is when because the binding of one transcription factor happens, it can influence whether the binding of another nearby factor happens or not. And we have a way of modeling that when that influence is only to itself and not to another factor. Obviously, you could come up with more complicated models. And this has been useful for other things, which I'm not going to go into. Okay, so all of that is actually relatively standard. It's not horribly standard, but it's relatively standard. This next two sections are more kind of interesting. So what we want to do here is now ask, if I change a single nucleotide, how much does that single nucleotide change this kind of readout, the binding of transcription factors? So you can actually do the model, fine, you can take your DNA sequence, you can make a single change. You then need to have a way of summarizing the effect of that change. So for example, right at the start, we use the absolute difference in the unbound state as our model of the change of binding. Actually, we found that a bit unsatisfying, and we've now used the relative entropy of our changed DNA sequence to the reference, but summed over all positions in the window. So when you make a single change, say at base plus 50, you don't only look at the vector of numbers at base plus 50, you look at the whole vector of numbers, such that if that base has changed the affinity of a binding site, that makes it less strong, and then that means that nearby other things can bind, you've summed all of that effect from that single change.
What's the size of that window?
So we changed that window size.
Now the order of hundreds?
It's usually 500 plus or minus or something like that. So this is the plot now of the change, this kind of summed relative entropy change by position. And each letter here, you can see that they're A, T, G, or C. There's a real set of letters here. Then at each position, there are 3 letters, which are the 3 possible changes. And you can see that there is it's a kind of a line that jumps around. There are also these really interesting high outliers. And in these cases, in most scenarios, what you're doing is you're creating a binding site that previously wasn't there. And then that strong affinity binding site is not only affecting itself, but has a kind of domino effect on the layout around it. So it not only affects the fact that it's binding here, it suddenly changes the possible different configurations nearby. And furthermore, you can see just visually that for this particular guy, more things have an effect around, actually, absolutely around the transcription start site than elsewhere. And you can argue that this is just one, and of course this is not. What we want to do is now we're going to take that, we're summing this now across all human transcripts. Now Michael and I always have a debate about whether he plots the lines the right way here. So this point that goes from here, this is minus whatever it is, 700. It goes up, 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 and then that joins to here, and goes over here and up here, and then that joins to, bloody hell, here, and then it goes back down again. I personally think this is the wrong way up myself, okay? But, you know, you do it different ways. What you can see here is that the kind of aggregate change in binding here has a hump near the transcription start site, and in particular there's something really fascinating happening around minus 30, which people can get excited about, and then it's generally, it falls away as you go away from the transcription start site. Now just to reassure you, if I go off and I add 500 base pairs to all of my DNA sequence, yeah, then all this shape moves by 500 base pairs in a particular direction. So this isn't some artifact of the fact that this is in the middle of our DNA sequence or anything silly like that. This is a real property of the DNA sequences underneath. And just as another control, what we can do here in this model is we can take all these states in the petals and we can now shuffle them. So we take our 70 transcription factors and now we build entirely new position weight matrices just by saying, oh, well, the first transcription factor will actually have, the first state will be the 5th state of this transcription factor followed by the 12th state of something else followed by something else. So we completely shuffle the set of states, maintaining all the affinity, the individual base affinities of those transcription factors. And when you do that and you run through the same procedure, of mean binding change, you can see that you get something and something which is very nice and flat, and that, which is sort of 2.4.
You're shuffling edges in the hidden archive model?
So we're sort of shuffling states. So we're shuffling emission probabilities. And you can see that that 2.4 is way below here. Okay. Now that shuffle, annoyingly, of course, won't maintain CPG bias and stuff like that, but it does maintain the kind of total affinity bias of the whole system to this. So that's that. And now we can do another business. We can take all the changes that we observe, and then we can look at the changes all the changes that we predict, and we can look at the changes which we have either observed that have changed over evolution, so they have been fixed in evolution, which is green, or the ones which are unobserved for a particular distance. So this is chimp, rhesus, dog, above is CPG islands, and below are CPG deserts. And what you can see here is that the observed changes, in particular in dog, particularly in CPG islands, are consistently below the unobserved changes. Now that's what you would predict, because evolution in aggregate favors neutral changes. So a neutral change will just fix at a particular rate, at its own rate, whereas a change which is deleterious will hopefully, well, usually not be fixed. And so the fact that we can sort of separate out the observed changes from the unobserved gives us separation is sort of reassuring. Okay. So having, I hoped, given you some reasonable reassurance that this model, so this is now a model of binding followed by a way of assessing changes that we're doing something sensible, we can now use it to ask some biological questions.
Are you going to say anything about the -30 oddness?
Oh, yeah, but that's the Tata bar binding stat.
Is that what it was?
Yeah.
And the changes were?
Yeah, there is something a bit weird, that it's slightly masked by this, that there's a second kind of dip here. You can't see it in this graph. I have some other graphs.
Up and then back down
It
again.
goes up and back down again. And this is all wrapped up into, dear God, GC, CPG content, and where your CPG islands are, and all of this kind of business. So this, though, is your expected sort of Tata box.
Tata, or you don't have a Tata.
Well, it's more that because so many, these plots are slightly misleading because you're registering around the transcription start site. So in particular, the Tata binding site, which has a very nice fixed distance, gives you a good, sharp readout. Whereas perhaps other things that have more variable distance, when you aggregate them, they don't really, they kind of fuzz out, basically.
Is
Yeah.
the position of that hump species independent? Or?
We haven't done that, but we would predict it to be. I mean, it would be kind of interesting if it wasn't. Let's put it that way. Okay. So now we're going to use this model to ask some hopefully interesting biological questions. And so again, for the audience who hasn't met this, very quickly, it's kind of miniature crash course in evolution. So mutations happen randomly, and most will exist at some rate in a population. So it's not the case. Nearly every mutation happens in a population at some rate. Only the mutations which are immediately lethal don't exist. So even mutations which are not particularly good for the organism, for the individual, so mutations which really make them not a particularly fit individual, they will still exist in the population. The way selection works is it either suppresses or increases the frequency of mutation over time. So a mutation can be neutral, meaning it doesn't matter, or it can be under negative selection, meaning that the offspring are less fit than the total population. And in that case, over time, the frequency of that mutation will decrease, or it will increase because of its beneficial positive selection. And there's also a rather large random effect here that just things randomly move around in a random walk. So over time, if you have negative selection, you observe less changes than you expect because selection is removing potential mutations and not letting them fix and then positive selection, you observe more change than expected. So positive selection is when there's a beneficial mutation that makes some effect that means that the kind of species evolves. Oh, it's not DA, DS. And there's going to be a couple of different things that we're going to talk about here. So one thing is that this rate of mutation is not the same across the whole genome, just slightly annoyingly. It varies by enough that it's irritating. And so this localized mutation rate, that shouldn't say DA, it should say DS, sorry, is often measured by something which we call DS, sort of rate of change in synonymous codons, which are ones that are thought to be neutral. What traditionally people have looked at is the rate of change of the acceptance of mutations, so over evolutionary time, of amino acid changes, and sometimes that's called DN, and so one gets a ratio DN to DS. What we want to create here is a new kind of parameter, which would be the rate of change in promoters, and we're going to call that DT, T for transcription. So we want to create this sort of DT number using our model. So that's is trying to say this. DN, DS are just special cases for how a genetic changes affect phenotype. DN is very kind of obvious, where you sort of just say that if it changes an amino acid, we just say it's a change of 1, and then we're going to just add that all up. So using this model, we can try and do this on promoters. And you can see Michael generally doesn't talk to mathematicians, he talks to biologists, and it's about as many symbols as you can get onto a slide without having people leave the room. So this is not particularly sophisticated at all. We're going to take this change parameter for every position, and there are 3 possible nucleotides we can change to, and we're just going to sum that over the whole promoter to have a thing that we're going to call big T, which is the kind of inherent changeability of the promoter. So not how much the promoter actually changes, but how much the promoter kind of could change on average across all the positions. And one interesting thing that we discovered early on is that this big T is not evenly distributed. So certain classes of genes have different inherent changeability. So this is for low T, which means that it's not very, it doesn't seem to matter so much when you change it. Now already you get different classes of genes enriched in low T. Now admittedly this could be for a whole bunch of reasons, and one of them is that we only have 70 transcription factors and there should be 2,000. So maybe we just don't know the right transcription factors, and so our model is just predicting the wrong thing. But one can sort of argue oneself that there's perhaps something else going on here to do with the fact that, as the biologists will know, that all of these guys always come up in positive selection for the protein coding genes. So it's quite weird that positive selection for protein coding genes, they also seem to have promoters that are very sort of uninterested in change. Now, the high T number is perhaps far more robust, because even though we only have 70 transcription factors, it's very hard to work out how other transcription knowing the fact of other transcription factors will somehow lower your T. So the fact that there's more complexity in the system, it seems very unlikely that more complexity will somehow precisely balance everything out that one would get with a lower changeability. And so this list of cases is much more interesting, but notice that there is quite a lot of kind of cell cycle-y, kinase-y stuff going on here. So lots of kinase-y sort of things happening. Remember, here we're looking at the promoters of genes, not at, for example, enhancers. So the region's very close to the promoters. So having come up with this kind of measure, so this kind of large T, which we're going to use down here, then we do a very simple thing, which is to create the observed difference for a particular, say, human to dog. We take all the observed differences and we just sum up those changes. And then we transform this. This is the Jukes-Cantor transformation, which just tries to take this measurement and handles the fact that one can think about reverse mutations as well. And so this transformation is again relatively standard, or it's very standard, about how one transforms this distance measure into a sort of rate. And so we now have a dt measure, and this is scatter plotting dn against ds and dt here against ds. You can see there isn't a strong correlation of dt to ds, and certainly less than dn to ds as well. And this is, don't worry about that guy, basically. And just so that we, create a new symbol, it's kind of exciting. So in lots of bits of positive selection things, this dn to ds ratio gets quoted. Sometimes it gets the letter omega. So very excitingly, we have decided to have the letter psi for the thing called dt to ds. There's no real reason why, except that it's a bit easier to write down. And so this is this psi number now, which is the sort of ratio of how much change is happening in promoters to its neutral rate. It's kind of nicely humped around 1. It's a little lower, which looks all well behaved. And in particular, it's not, this is psi on the left-hand side, and this is big T now, which is the inherent change on the x-axis. And it's not correlated with this kind of propensity to change. So what's driving psi is not obviously the fact that some promoters are more changeable than others. So these are the observed differences on this side, or the ratio of the observed difference versus the local background rate. And then this is the propensity to change from our model. There isn't a strong correlation there. And there isn't really a correlation so much with omega either. The fact that there aren't anything in here is apparently, this is a PAML-ish issue, PAML bug. Apparently it's a known PAML bug that PAML doesn't like to predict those omega values. And these points over here, I think, also are PAML bugs as well. So it's basically flat again. So this is the protein rate versus this promoter rate. There isn't a great deal of correlation. You could argue that this tail is going up a little bit here. So having taken the psi, we can now do the classical thing for selection, which is now you can either rank genes or more perhaps appropriately do Go lists of gene functions. So these are Go categories. I've shown some of them already, but you can group genes into broad categories. And this is for low psi. So this is where we're saying that these genes are under negative selection. Their promoters are changing far slower than we would predict against the bulk. And right at the bottom are the things that you would probably expect. So multicellular organismal development, developmental process, all of these sorts of things. These promoters are completely locked down. They do not want to change. This is unsurprising. These are presumably very complex genes, but it's a sort of an unsurprising, reasonably unsurprising result, I guess. And then this list of things here, again, basically are reasonably well predicted, I guess. It's all about complex transcription and stuff like that. So this is a reasonably standard result. This list is the other set. So these are the genes which are changing their promoters, that they have more functional changes in their promoters than you expect. So the categories that are functionally changing their promoters faster. And this list is not agreeing with any particular prejudice that myself as a biologist has. So then you have to sort of settle back and decide whether this is interesting or not. Certainly this top one here, RNA processing, is a little bit odd. And then down here, sometimes you can get excited about this and sometimes you can't. We get excited about the Golgi vesicle mediated transport and Golgi vesicle transport here being kind of lipid trafficking sort of stuff going on in maybe complex cells. And there's some other sort of things down here. But this list is not your obvious list for this ratio. And so this is just showing the distribution of that Golgi those Golgi things against the background distribution, just so you get a sense of what it looks like, and it definitely does look right shifted. Michael wanted me to put this in, that this sunflower model is a software package that people can download and use, and other people are using it now. So if you did want to do this and run this yourself, you could do this. And Michael is expecting to give this out in April. So this is the conclusion. We've got a model of TF binding, quantifies our potential mutations, affect transcription factor binding, and potential positive selection and purifying selection in promoters. Now I'm going to end here with a couple of kind of questions or statistical questions that I feel we haven't really grappled with in this business. So it's a bit annoying that relative entropy is not symmetric. The prejudice would be that we want to have a kind of symmetric score about the difference between two probability distributions. And it's a little bit irritating that it's not symmetric. We don't worry about that in the moment. We just say, this is the reference and we're changing it to dog. This is the reference, we're changing these mutations. But that's kind of an annoying process. And also, I don't know if you've kind of noticed this, but we often add it, okay? And it's not obvious to me that adding it is the right thing to do. So we take this and we have 17,000 cases, and then we sum those 17,000 cases. We get what I think are reassuring plots and distributions and things like that, but it's not obvious to me that that's the right thing to do. So if anybody's come across a problem about, different sets of probability distributions and what one should do and whether one should add on them or not, I'd be really interested. For the biologists in the room, I find it really interesting in this study that we had to create this thing which we call big T, which is the inherent ability to change, for the amount that the promoter itself could change. We don't often talk about that in the NDS. We don't often talk about the fact that proteins must have different inherent sensitivities to mutations and things like that. And so should we talk about that? Is that a useful thing or not? There are some people who do this, but it's not kind of mainstream in, I think, in current ways of thinking about protein selection. It's kind of interesting. This one, I think we have some really good ideas. If you want to grab me or someone else for the Blackboard, we have some ideas. I don't know if you noticed, we only deal with substitutions here, okay? When you have indels, life just becomes so much more complicated and so much more fun. But this is a kind of complication and fun that I am well used to now. So there are some ideas about how we're going to do those, and I can go into them at some point. And the final thing is this annoying CPG effect, okay? This annoying business that everything seems to be kind of keyed off GC content CPG. I just want to spend a little bit more time with that. Just to remind you, this was this very simple plot. We put the GC content of the predicted affinity here on the y-axis, total binding on the x-axis, and clearly there's a huge correlation of this. Up here you have a factor like SP1 that's known to bind everywhere and be a kind of general factor. Down here you have a collection of like FOX D11 and SOX9 and GATA3, all of which biologists would know as their very specific targeted transcription factors. And this is this general problem about heterogeneity, and I think Peter will talk about it more, and it's one of the real headaches to genome analysis generally, and I still do not feel that we do this right. So there's a lot of things in the genome, and it's lots and lots of things in the genome, and they're correlated to each other a lot of the times. And so there is a kind of rule in genomics, everything is correlated to GC content. It doesn't matter what thing you're measuring, if you plot it against GC content, you will always get some kind of correlation. And that's partly because GC content itself is correlated to gene density and exon density in the genome. But the whole thing's kind of confounded in there. No matter what you do, you've got to handle it. And we did it by splitting the genome into two classes. So CPG island promoters, non-CPG island promoters. There is some biological correctness for doing things. There really is a binary distinction. You're either methylated or you're not methylated at a single CPG. in the germline. So there is a real kind of split there. But that's for a single CpG, not for a set. And anyway, it's a bit ugly to just pretend that the whole genome is split into two separate classes. Sort of slightly more irritatingly, from my perspective, is that the biological machine itself may utilize this split. It's not the case that the right approach is just to sort of remove this problem. So that goes back to the fact, going back here, that SP1 is highly GC rich, predicted. It binds very highly, but it binds also on lots of housekeeping genes, which also have CpG islands. So it's not really fair to kind of penalize SP1 for being so GC rich in its binding. It's doing the right thing. It's finding the promoters that it should bind. They're GC rich. It's got a GC rich binding site. What's the kind of big issue here? So I'm not quite sure if the right thing statistically is to try and remove all of this kind of correlation, as it were, that's going on. So I summarize that as what's our null hypothesis that we're testing, but I'm sure there's more sophisticated ways of stating what I've just done. And then this comes back, it's sort of similar to the null hypothesis business, but if you notice, we had a state called the unbound state, and I didn't tell you how we parameterized that unbound state. And in fact, it was just the background frequencies of A, T, G, and C across the whole genome. Now, should we have done that? Should we have had two models, one is CpG island and one is not CpG island, and put background frequencies there? Should we do something else? If you're a biophysical modeling kind of person, you say, well, what we've got to do is have histone binding stuff and things like that also in this process to understand this background. But that's, I mean, to be honest, you're never going to have enough knowledge to really produce the full model. So what should we do with these states when there is such heterogeneity in the genome? Should one change your background models or not? So those, for me, I'm perpetually grappling with this, and I don't feel that there's a consistent way of handling these sorts of problems. So just to stress, most of this work was done by Michael Hoffman, who's currently touring the States, and then there are many other people who have contributed, and Michael was mainly funded, in fact, by the National Science Foundation in the US. So that's my talk. So thank you very much. Yeah. So I think we got time, 10 minutes for questions. Julie?
In the last part, you talked about, first you told us about transcription factors and if they were sensitive, predicted to be sensitive to change or not.
Yep.
Then you talked about promoters, the type of promoters that actually do change.
Yep.
Or don't. And is there any correlation between those two?
That was this big T to...
I missed that.
That's okay, it's a good...
Because they didn't look like they were the same risk.
No, and so it's this plot here. So this is the observed change factoring out the local mutation rate. So dt by ds on this axis. And this is the propensity to, our predicted propensity to change on the x-axis here. So that you can see that's a reasonably flat scatter. So it's not the case that the ones that are changing a lot, that we predict are changing a lot, are due to the fact that they just have a higher propensity to change.
The ones that are...
So the set that we observed which are changing a lot, which is kind of up here, or this, you draw a line somewhere on this psi-axis here, come from all classes for the propensity to change. So there isn't a correlation between the propensity to change and the amount of observed change that we see.
Yeah. But then if you overlaid on that the Go terms for the systems that are involved, I bet you will see a pattern. Because I was going to ask you, you pointed out the well-known result of immune systems having low propensity to change.
Well,
did you find the positive selection signal for such systems?
So just to say, the immune system in this plot would be over on this side of the axis here. We see it in the x direction, but not in the y direction, and it's so it's well known that they're under positive selection for their genes, their DNA sequence. Quite how to interpret the fact that they're over to the left side of this plot is hard to know. And certainly you could argue that a better way to visualize all the Go categories is to plot it something like this, just to get a sense of these two axes here, rather than those lists. There's quite a lot of different ways you can cut this data. So the interpretation of what's going on there, I don't know. So one interpretation is very simply, we do not have the transcription factors that underlie the immune system. And if we did, they would be in here and up here. Yeah. So I agree that we can't say anything about that. But the stuff which is on the right-hand side of this, I think we can be relatively confident about. Because it's quite hard to work out how, by adding more data, you end up with less complex promoters. By adding more transcription factors, you'll end up with a less complex set of promoters. So the things which are on the right-hand side of here, which are mainly kinases, interestingly enough, I'm more confident about that they must be on the right-hand side of this. Wolfgang or Ed? Ed?
So what I'm wondering is how much would the inclusion of copy number variations possibly change those results?
So of covariations or?
Copy number variations.
Oh, copy number. Oh, Jesus. So we're because we're dealing with 1,000 base pairs each side. A copy number variant would presumably take a whole chunk and duplicate it, and that's not in our model at all. Are you talking about copy number variants of the transcription factor or
of the DNA regions? But it could be smaller.
Well, so yeah, at that level, I don't call those copy number variants. I would call those indel events. And indeed, working out what on earth you do with indel events, that's another 30 minutes worth of chat, and it's not obvious how one does it, obviously, but I can imagine some ways of doing it. Ed?
Just on the scalability of your model. So you have 70 transcription factors
Yeah.
now. If in the limit you actually have the
It's fine.
for all 2,000, you have 2,000 transition probabilities out of your start point. And,
Yeah, so there's no computational headache. I mean, it's not the easiest thing. Remember, we don't train the affinities, okay? And Martha's not here, but one of the reasons to get Martha here is I think it's so great that there are biochemical ways now to really measure those affinities directly, and that's that's going to change the game so completely.
yeah,
So,
but they're going through most of mouse right now, right? So
yeah, and frankly, I think mouse is. I mean, we can flip this all over to mouse if we so wish, but I think the affinities that we measure for mouse will be just the same as the affinities for human, give or take one or two cases. A more interesting question is what on earth to do with these transition probabilities for the concentration. So you could set that by gene expression data, with some scaling, but then you're doing it only on the lymphoblastoid cell lines. And you want to do this in some way across the effect of all cells across the organism. So God knows what one should do.
something like that. So you might simplify it.
Yeah, I just Summit. I don't know. I mean, certainly there's n parameters for the number of transcription factors that we just have to kind of set at the moment, and that's not particularly pleasant.
How sensitive is the model to that setting?
It's not. I mean, so it's not horribly sensitive when you set some of them to 0, because we've done that. But we haven't done something that's systematic, and probably we should do something that's systematic just to check how all of these things vary. So if you start varying that number around, those numbers around, how it works out. Elliot?
So just to extend this a little bit, it's interesting to think about how you may want to develop this algorithm across all cell types in an organism, which the DNA is constant. But if you do want to focus in on a particular cell type, it would be interesting in the future to maybe take into account both gene expression, I guess, of the proteins, or ultimately protein, some sort of protein measurement, and then some sort of chromatin state.
Yeah. So we tend to when we're looking at this, we tend to use open chromatin maps. We don't actually look at the whole genome. We just say these bits are open, everything else is boring. Okay, it's switched off. So certainly, it's a rather kind of polarized on-off view of the chromatin world, but one that I think is fine to take. But Aaron Segal in particular has these models, which is very similar models to this in some ways, but where he's trying to very deliberately model the gene expression to fit observed data. So he actually learns those parameters, and then you can think about this. He doesn't have a time axis, but you can think about then modeling this with a time axis with a feedback loop, so that the system evolves over time. That's even more complicated. It's bad enough just doing the learning of the parameters for this. But because we don't feel that our niche is in that area, our niche is in trying to assess the mutations. Sure. Yep.
So when you consider the transcription factor binding, you only consider very simple architecture in the sense it's very close to transcription start site.
Yes.
And also, my understanding, you only consider very simple motif in the sense you didn't consider those 2-block motif or bilingual motif. So do you think you consider more complicated architecture?
So certainly Sunflower is agnostic to the piece of DNA that you give it. You can give it enhancers, but what it's giving you a readout is of the binding affinities of enhancers, of transcription factors to the enhancers. So that's one aspect. There's nothing in the system that doesn't prevent, a 2-block motif, which is, I don't know, GTT, NNNNNNNNNNNN, TGT, or whatever, or backwards, going the other way. It'd be absolutely okay for us to put that in the model, but obviously you need to. We don't have any facilities for training a model from scratch. I mean, someone has to tell you that this is the truth. In particular, another thing is that what the model won't allow you to have is things that are binding inside of the buffer N sequence, which does annoyingly happen, but most of the time it kind of doesn't, and tough. We're just not going to model that first off. I think a more interesting thing to this is this cooperativity parameter that we've added. So this is where the binding of one transcription factor somehow influences the relative affinity of another transcription factor nearby. And so we do have that model. Again, you can argue that model is not perfect for certain reasons, but that's where we feel the model sophistication should come in next, is this cooperativity parameter.
Okay, the second question is, you present a numerical analysis, basically says, okay, you fix motif and you shuffle the motif matrix.
Yeah.
And you want to see, this is a kind of your control, and
yeah.
you want to see the significance of prediction. But that, you present the argument that says, okay, that make conserve the CPG.
So it conserves the GC, the total aggregate GC-ness of the set. It doesn't conserve dinucleotides, for sure.
Right, but I just feel some uncomfortable that fixed motif and the shuffle matrix.
So again, if there's a more, it would be interesting for us to think about other ways of doing the shuffling or whatever. So remember, the other piece of justification I said that the model works is the fact that when we look at human to dog differences, the unobserved dog differences are definitely, on average, making more effect than the observed differences to dogs. So we think that's another biological readout that is not in our model. There's nothing in our model that knows anything about evolution, and yet we are creating numbers which agree with this business that evolution generally fixes more neutral mutations than deleterious mutations. But it'd be very interesting to work out better ways to shuffle the model or to do something more sophisticated. I'm reasonably, I'm sure we can do better, but I'm reasonably happy about that end of the business, that we're getting a real biological readout that is unique to the DNA. Okay, shall we end?
So yeah, let's thank you again for this. And there's now a coffee break, and you continue at 12:30.